Next up, we have Maor Matrzafi, and Maor is coming from the Department of Plant Pathology and Weed Research at the Volcani Center in Israel. So thank you so much for joining us from outside the Northeast. We really appreciate having you here. And whenever you're ready, take it away.
Thank you very much, Eve. And I also want to thank Tony for the invitation to talk in this interesting conference. I very much enjoyed the talks that were before me and also planners that presented their subjects before that. I'm going to speak today about the topic of climate change and how it affects herbicide response. And due to that, the sensitivity of specific weed species, generally invasive weed species, but also others. And I think that if we look at the scenarios today, we already know that there is an increase in the number of invasive species around the world. The distribution is increasing, the damage is increasing. Here you can see several species that we have now in Israel. Amaranthus species, the Amaranthus tuberculatus, had invaded Israel a couple of years ago, but in recent years, it's getting more and more to be more and more common in agricultural fields. You can see also several solanum species that are also very abundant in our fields and increasing damage to minor crops, but also to major crops. And we can also see species such as Parthenium. This species has invaded more than 50 states or 50 countries around the world. And it has a lot of health issue damages also, and not only agricultural damages, since it is very allergenic. And we know that the flowers can give you a rash, the leaves can give you some kind of a skin dermatitis. It's a very dangerous species for human health. I prepared the table that showed several of the species, the most important species that we have. We can see the cypress, rotundus, purple nutsage. In some areas, we also have yellow nutsage. We can see the main habitats that they inhibit for now and the propagation way. And if you look at the list of the species, you can see a lot of them has propagation by such as the convolvulus, the sorghum alpense, and also the solanum species. Is not by seed, it's more due to rhizomes or root fragments can get established in more harsh environment, in more extreme conditions, and they lead to further damage in different crops such as orchard, vineyards, and also in field crops that we have here in Israel. And now I want to present some of the studies that have been done in recent years and talking about the effect of environmental changes and how they can change herbicide response in different weed species. So first I would like to start with one very interesting study that was done in Israel in 2016. It was published in my old lab. This is a PhD student from my old lab that worked with my mentor. And here you can see the effect of temperature increase on the response to glyphosate. Now, what we see on the y-axis is the 80-50, and the 80-50 represents actually the effective dose that we have that kills 50% of the population or 50% of the plants that were sprayed. What we see over here is 3 different populations for each species. And you can see that the overall trend is reduced sensitivity as we increase the temperature. All the populations that we have over here for the Conyza bonaerensis show it. Some of them are less responsive for that, but in Conyza canadensis, we see more response and reduced sensitivity as temperature elevates. Another very interesting study that was done in Mitila Jugulam's lab in Kansas is studying the temperature-dependent response of Amaranthus palmeri to mesotrione. Mesotrione is an HPPD inhibitor. This is a very interesting herbicide, a very new herbicide, and we can see that when we increase the temperature, we actually get less sensitivity of these Palmer amaranth plants to the herbicide. When we look on specific rates, you can see that at the low temperature, we saw very good control at 26.25 gram active ingredient per hectare. But when we increase the temperature, we can see that even at double of this recommended rate that we have over here, plants are still surviving. They look very good. the plant injury or damages that we saw in here at low temperatures are disappearing almost completely at high temperature. You can see some kind of an inhibition in growth, but these plants can still grow and produce seeds at the end of the season. Another interesting study that we did when I was in my PhD, we tried to take several grass weeds and see how the response is going to change when we grow them at different temperatures after herbicide application. So here we tried a herbicide. This is a ACCase inhibitor that's called diclofop-methy, and you can see here several herbicide-sensitive population or species that we tested: Alopekurus, Avena, Lolium, and Setaria, both winter and annual and summer annuals. And we also took as a reference as such a control group, Lolium population that is target site resistance for the same herbicide. And what we can see over here is when we spray them, and this picture we're checking 21 days after treatment, we can see that all the plants that were under C, this is the control, look very nice, and the plants that are under T are fully controlled, as we can see, for all different species, except of course for the target site resistant population that doesn't show sensitivity for the herbicide at all. Now, when we go for the next step and we tested them or we grew them after herbicide application under 34-day, 28-night Celsius temperature, we can see that there is an increase in survival at all species in the plants that were treated by the herbicide. In some of these populations, we got into 80% non-controlled plants. From 0 to 80% uncontrolled plants is a very high number, and we can see the reduced efficacy of the herbicide caused by the increase in temperature. Now, a couple of years ago, I've summarized all of this, and this is a paper that was published in Pest Management Science in 2018. Some of these cases are from this paper. We already have more cases that have been reported all over the world. But what is interesting to see here is the differences in species, in temperatures, how the temperatures affect the herbicide sensitivity or herbicide response, and also that we have the same trends or the same response also for sensitive and resistant populations. So it means that even if a population was already sensitive or is already resistant, it can increase the resistant ratio for that specific population. Now, if we look further into the different research that we have over here, then we can see, for example, in this paper, grass weeds, this is what I showed you, there is reduced efficacy under high temperature. In paraquat, a different herbicide than diclofop from a different mode of action, we can see that the sensitivity is actually increasing in reduced temperature. So there is an opposite trend between those two herbicides. We can also see a different trend between species. For example, these are Coniza species that showed reduced efficacy under elevated temperatures. And for Ambrosia species, we can actually see that glyphosate, we have reduced sensitivity under low temperature. So that means that temperatures play different role for different species and for different herbicides. And it means that we have a lot of things that we still don't know, and we need to understand about this mechanism of temperature-related herbicide sensitivity. Now, one more paper that was published, and this guy, Louis Ziska, has worked a lot on how CO2 is affecting herbicide response. This was done in the 90s. And from this work, we started an idea to try and find out what is the effect of the combined temperature plus CO2 effect on herbicide sensitivity. And this is what I'm going to show you next. This is going to be plant response to these two different parameters combined together. And what we tested is temperatures, normal temperature and high temperature, and CO2 level, the current CO2 concentration that we have, and the increased CO2 concentration that is predicted for the future. We can see here the response of Chenopodium album for different CO2 levels and different temperatures. The number that you have over here is percent of plants that survived the treatment, those treated plants in T versus the C, the control. You can see that there is an increase in survival of plants as we go through CO2 levels, but actually the higher increase is as we go through temperatures. When we combine those two together, we see that we had the highest actually percentage of plants surviving the treatment. So it means that there is some kind of a synergistic effect or combined effect between those two parameters that increases plant survival. We did the same thing for Conyza canadensis. It's more difficult to see here because this is a rosette leaf plant, this is an erect plant, but still we saw an increase in the number of survival that was approximately similar to what we saw in the combined treatment for the Chenopodium album. So after we saw this, we started to try and understand what is the mechanism of this reduced sensitivity. We tested the translocation of glyphosate in the leaves under different temperature and CO2 levels. And here I'm going to present Chenopodium album and Conyza canadensis and the amount of herbicide that we saw translocated to shoot meristem and undeveloped leaf and to the roots. Now, these two parts of the plant are very strong sinks, and this is mostly where glyphosate is going after you apply it on the plant. And what you can see here in the dashed line, I'm sorry for the small capture here. In the dashed line, you can see high temperature and high CO2. And in the solid line, you can see low temperature and low CO2. Now, we can see the differences. We have more glyphosate in high temperature and high CO2 going to the shoot meristem and undeveloped leaf. And the same trend was shown for the roots. Now, looking at this, we can think, okay, these plants are taking more glyphosate and moving it into those very sensitive parts. So why these plants are resistant at the end? And our hypothesis and what we try to model in here is the response and what happens when glyphosate interacts with the plant. So you apply your herbicide at the first day, and after 48 hours, you can find it, as we saw, in strong sinks all over the plant, young leaf and upper meristem, and the roots that you see over here. What happens after that is after 7 days, the glyphosate is actually controlling those areas very specifically. As you can see over here, the plants, the leaves that they have are actually controlled. And we see after 14 days, regrowth of the plant from lateral buds. What happens for our model is that herbicide actually impairs its own translocation from those areas, and other plant tissues can regrowth and regenerate, and this is what we see over here. We saw this phenotype also with our plants, as you can see them 70 days after application. For the treated plant, we saw a lot of tillers, a lot of buds that are going, starting to grow from lower parts of the stem. It gets kind of a bushy phenotype in comparison to the control, as you can see over here. In the Conyza, we also saw the same thing. We saw multiple regeneration of shoots, but as you can see over here, it's more spread through a lower part of the plant. And instead of this plant, the control plant that shows a very normal phenotype for Conyza. Now for our research, we actually finished in this point, but in other papers that were published in different labs around the world, we saw these trends for different herbicides. We saw reduced absorption of glyphosate for Bacillus scoparia, and we saw also rapid metabolism of 2,4-D for Amaranthus tuberculatus. And those conditions of temperatures is actually increasing or reducing the sensitivity of the herbicide. We know over here that it's the reduced absorption. The plant is absorbing less herbicide, as you can see, under high temperature. And in here, you can see that there is rapid metabolism. When we look on different time points after application, we can actually see that in the red bars, the high temperature, we can see less of the 2,4-D metabolite in the sensitive population and also in the resistant population. So the detoxification processes are enhancing through the plant in a higher temperature when we apply the herbicide. After we look on these studies that I've showed you, I think that it's pretty much obvious to say that there is reduced herbicide sensitivity that is responsive by temperature, increase in temperature, elevated CO2, and the combination of both factors. Now we saw that the combination is actually getting more resistance or reduced sensitivity than each of the parameters alone. As we look to the future, we know that these two parameters are going to be increasing together, and we're not going to have only increase in temperature, but we're going to also have elevated CO2 levels. Now, we can see that the mechanism, the physiological mechanism, is related sometimes to the reduced absorption or translocation of the herbicide to specific tissue, some kind of a sequestration in specific plant areas that reduces the herbicide efficacy. But everything is on the basis of breaking down the herbicide, impairing the herbicide, and reducing its efficacy all over the plant. We need to think about reassessing our current approach to weed science. The weed control era that we have now is mainly dominated by herbicides. We have to take on proactive climate change-driven approaches. If it means using more IPM, using more mechanical approaches, biological approaches, and other things, but also we can think of ways to improve our chemical activity by using all sorts of inhibitors for detoxification processes. We know that some chemicals can inhibit detoxification in plant and can increase herbicide translocation, maybe move it more safely to the specific site where it should move by encapsulation or other methods that are being developed in the chemical world. So using all of that, I think that we should think more clearly about the future of herbicide application. There are a lot of things to still study, and we are still doing several more studies about this subject, hoping to give more results and clear up some points in the future. At this point, I want to say thank you to all of you for listening to my talk, and if you have any question, I would be more than happy to answer.
Great, thank you so much. We do have a couple minutes for questions, so if anybody wants to enter anything in the Q&A, but I do have one question that was sent to me over the chat. So would you expect the same issues or patterns in woody plants compared to the, the herbaceous species you've been studying primarily?
Yes, so this is a very good question because we know that movement of herbicide in woody plant is a little bit different than herbaceous species. But generally, yes, I would expect less responsive of herbicide in those species. I think that generally, when we look on the physiology of the plant, increase in detoxification processes can happen in herbaceous and also in woody species. It's not something that is specific for herbaceous species. The translocation could be a little bit different because we're talking about systems that are more oriented through translocation in vascular systems that are a little bit different, but generally I think it can happen also in woody species.
Great, thank you. So no other questions have come in through the Q&A, so I would like to ask you, so where do you see yourself heading? Do you think you're gonna keep doing more of these trials or start thinking about how we can change kind of the mechanism for how herbicides are applied? So I guess what are your next research goals?
Okay, I think that general, we still have a lot of issues that we haven't answered yet. For example, we don't really know what is the time frame that is needed for detoxification of the herbicides. If we're looking at the processes that happen, regularly, we can see that in season, we can have 2 or 3 days of extreme heat wave, and after that, we go back to the temperature that we had at the beginning. So we still have to test if 2 or 3 days are long enough for detoxification of herbicides. How much time would it take for each herbicide to be breaking down or impaired in different plant species, and generally do some more phenotyping of the phenomenon in different species. As I showed at the beginning, we have a lot of variation between species and herbicides, and there are things to be shown and studied in this area.
Great, thank you so much. And there was one more question that came up in the chat just now, so if you wouldn't mind taking a look at that, that would be awesome. But thanks again for joining us. This is great to have you.
Sure, thank you.
And if you wouldn't mind, great.